Ready to go. Thank you so much for taking the time, and thanks everybody for waiting.
Yeah, awesome. Yeah, thank you so much for having me. Thanks for coming out on a Wednesday to learn about sharks. I am Stacy Farina from Howard University. And just to give you a little bit of background, I did my bachelor's at the University of New Hampshire in marine biology. I have a PhD from Cornell in evolutionary biology. I did some postdocing at Harvard, and I also was an adjunct at Northeastern University in Shoals Marine Lab for a few years. And currently, I'm an associate professor at Howard University. I just got tenure recently, so I'm excited to finally, make that milestone. Feels good. Right? Yeah, exactly. Now it's just smooth sailing. I can do whatever I want. Unfortunately, not so much. But we have fun. So in my lab, so that's what I'm doing at Howard University. My focus is actually, so this is my big secret. I'm not actually like strictly a shark biologist. So most of my work in the lab is on ray fin fishes. That's what all my graduate students study. But I have done some cool work, especially with former students from Cornell and other opportunities that have led to these cool shark projects. So that's what I want to share with you because I wrote this talk for Shark Week. Unfortunately, that event was the evening event was canceled, and so now I'm here to talk to you guys about sharks. And I hear that you're really interested in especially sharks, shark teeth. So let's get right into the shark teeth. So I call this mysteries and myths because I really like doing research around sort of misconceptions or things that are kind of taken for granted that we already know all we need to know about them. And one of those is the fact that sharks continuously replace their teeth. It's a very common piece of shark knowledge to have. And so you can see on this tooth whirl here from a great white shark, this is a CT scan, that sharks continuously replace their teeth throughout their lifetime. And they grow their teeth in this kind of array where they have developing teeth ready to go and replace the functional teeth. So they can have a few functional teeth at a time, and then they've got a bunch of backups at various different stages of development in the jaw ready to go. So I'm sure you've seen that in the shark jaws that you've looked at. You can see it even in preserved tissue. And shark teeth are abundant in the fossil record. As you know, you're interested in fossils. That is a very common thing to find in the fossil record are shark teeth. They fossilize really well because they're made of enamel and dentin. And the rest of the shark skeleton, unfortunately, is not. And so because most of the shark skeletons are made up of cartilage, there are very few sort of fully articulated, fossilized shark skeletons. And whereas we know many species of sharks, extinct sharks, only from their teeth, because the teeth are the microfossils that really fossilize very well and give us a history of sharks that we can't really get from the rest of the skeleton because it's very difficult to find. So that's just a little bit of background on tooth replacement, but my mystery that I want to present is, are shark teeth single use? So we know that shark teeth are replaced. We can see, there's been some tracking of shark teeth a little bit in aquaria, and you can figure out like the rate at which teeth are shed. But we wanted to look at the functional side and ask how quickly teeth wear down. So to kind of get at the idea of how many times can you use a tooth before it starts to wear down and lose its functionality. And so to do this, we had to develop a dynamic testing system. And when you do testing of biomaterials, you can do what's called static testing, where you might mount it into a machine called a materials testing system, where it slowly lowers, let's say, the tooth into maybe some sort of prey item and measures the force as the tooth is penetrating. There's been lots of that kind of work done, but really that's not a realistic depiction of what shark teeth can actually do. You need dynamic testing where the physical conditions actually match the reality of what the sharks are experiencing. And so we know that when sharks bite down, many species do a rapid head shaking to have the teeth shake that move back and forth in a sawing like motion. We also know that if you test a shark tooth individually in a static condition, That's not necessarily representative because teeth are, you can see like this shark jaw here, there's many teeth interacting with the prey item all at once. And also bite force. So you have to mimic the fact that the shark is also biting down. So it's not just the capability of the tooth to penetrate the material, it's also about how the shark is applying a force while that's happening. And so you can imagine that a system like this is going to be very difficult to develop because there are a lot of different factors. You have to move the jaw very quickly. You have to have all these different things controlled for. And the reason that I have Dr. Lisa Whitenack up here is because she's actually in the process of developing such a system. And she's presented it recently at a couple different conferences and shown some really promising preliminary work. But we did this work back in 2015 is when we actually collected the data. And so we didn't have any of this. So what do you do when you don't actually have, a fancy testing system where you have all these things controlled? You glue teeth to a power saw. That's what we did. Yeah, why not? We love power tools in science. So we used the system because it we timed it so that we did some calculations to figure out that the reciprocation of the saw was actually very similar to the frequency of head shaking in sharks. Also, the power saw was able to provide some weight that mimics the force of the shark pressing down. So we got the bite force, we've got the head shaking, and we also were able to mount multiple teeth onto the jaw at once. And we had it going through a prey item like salmon that would be a somewhat realistic prey item that a shark would eat. And so many thanks to, the person who led this, so Catherine was an undergraduate at the time. She's now an assistant professor at Washington State University. She just started, but there's her with her power saw and ready to go. She really loved this project. It was a great undergrad project. And Jeff Bash is the head engineer at Friday Harbor who designed this whole very nice counterweight system where we could just release it and have the saw fall onto the prey and the weight of the saw would apply the bite force. And it worked out surprisingly well. We were all really shocked that this actually made a very nice system for us to test these teeth. So here's a nice glamour shot of the jaw after cutting through some salmon. And we also had it kind of set up so that it was like kind of food grade. Like we had cutting boards that we sanitized and everything so that people could actually eat the salmon after. I think Jeff was the one who wanted to eat it. But you can't have that much salmon and not eat it, right? But here's a video of the saw in action. And let me just say, the videos themselves were we collected data from high-speed video, but the quality was kind of not great. So this is just a regular video, but it gets the point across. So like for taking our actual measurements, we measured how much the saw was descending through the prey as it was descending, and that was our and so basically the rate at which it was able to slice through was our main thing that we calculated as our response variable for the experiment. And so we had to slow this down quite a bit to be able to see all of that and calculate that. But this is what it looked like to the naked eye when we were just kind of doing our experiments. So here's the video. So the answer to the question, are shark teeth single use, is actually it's complicated. So if you look at the tooth at the black here, Parkerinus falciformis, it performed very highly at first, so it really just sliced through that salmon like butter. And then after we did repeated trials, it ended up kind of losing that initial capability that it had. And we also saw that in the tiger shark in the red there, we saw an initial high performance and then kind of a drop off and a steadying of performance. And then we also saw with so we did a 6-scale as well. And we found that the 6-scale was pretty much very consistent in the, basically no matter how many trials we did, the tooth never showed a difference, although you can see that it was the lowest performing species. So what we can take away from this is maybe some species, like the first two, are have teeth that may be single use. You can see that it starts off really high, and then after, that's one prey interaction, and then after a few more prey interactions, the teeth are worn down. They're not able to cut through the salmon nearly as well. Whereas in the sixth gill, even though the performance was much lower, we were able to, we never saw a drop off. And so potentially there are some teeth that are single use in some species, and in others, the teeth are maybe longer term, although maybe lower performance overall. And that tracks because sixth gills, they have a very low metabolism, they eat very large prey, and so potentially they are not going to be constantly replacing their teeth. Their teeth probably work well enough for the kind of prey that they're eating. All right. Any questions so far?
So you took some teeth that were further down in the mouth in order to get fresh teeth to start?
So,
I mean, you can't.
yeah. So I think we took, and this would be a question for Catherine. I don't know if we took the functional teeth, because the question is, so had the teeth already been used? I don't remember if Catherine actually, or if we decided to actually go that route. It was so long ago. But thinking back on it, probably not. So that would be a confounding factor for sure. So some of these teeth may have already been used in the wild.
And then when it's actually shaking its head, depending on where in the mouth those teeth are, is whether they're going sideways or the shaking is going perpendicular to the teeth, right?
Yeah, so there's teeth in all different orientations. So this is kind of an artificial, having them all in a row is not necessarily reflective of how they're going to be oriented in the job. But this was the first study that tested multiple teeth. And hopefully, Lisa will someday have her really fancy system where she's able to control for all these things. But for kind of like fish summer camp, this did pretty well. And it's an indication that these teeth do wear pretty quickly. Especially, we can see a drop off in performance very quickly. And so we know that the teeth wear down more easily than we might have predicted.
Is there a particular pattern of wear on the teeth?
That's a great question. Yes.
Or something else?
So these are the before. So we took pictures before, but if you looked at the after, there were a couple just critical failures of the teeth. No failures of the glue, thankfully. And so we had teeth where the top just cracked off. And that was probably when they were hitting like the spine of the salmon. But we didn't do any like microscopy to look at the details of the wear pattern. Any other questions? Great question. All right, awesome. Okay. All right. So now this is more of my bread and butter. So that concludes the shark teeth portion of the talk, because this is my real passion in life, is studying fish breathing. So most of what my lab does is we study how fish pump water over their gills. And we've also done some cool projects on sharks that I want to share with you. And one of the biggest myths that you guys might be aware of as a group that are really interested in animals, that this is a myth, but this is one of the most common things that I run into when I start talking to people about shark breathing, is they ask, don't sharks need to constantly swim in order to breathe? And so that, just to orient you, that swimming to breathe behavior is called ram ventilation, where a shark will swim through the water with its mouth slightly open, or in this is a filter feeding basking shark, just to show you the mouth is probably open wide for filter feeding, but you can see that the mouth is a little bit open, water flows in through the mouth and out through the gills. And that's very important because when you look at the blood flow across the microstructures of the gill tissues themselves, the blood is flowing in the opposite direction of water. So being able to push water directly over your gills forces water to move in the opposite direction that blood is flowing. And that creates what's called countercurrent exchange, where you have the freshest water with the most oxygen interacting with the blood that's about to leave the gills and go to the rest of the body and is able to take up just a little bit more oxygen. And then by the time the water has gone through the gills, it's interacting with blood that is just coming from the back from the heart and needs to pick up oxygen, and it's able to pick up just a little bit more oxygen out of that water before it goes out the gills. And so this is a very efficient system. If these two flows were in the same direction, what would happen? Exactly, yeah. So you get to 50% capacity for both, and their exchange would stop because exchange of gases, this is passive diffusion, and so it's driven by a concentration gradient. And so if you don't have that gradient, you don't have exchange. So ram ventilation can be very effective for that. Yeah. Mhm. The gills are 100% artery. So blood, it's a single circuit in sharks. So an artery would be a blood vessel going away from the heart. And so in fish, you have heart, gills, body, heart. You don't have like an extra circuit. And so you're going to have basically all blood that's coming away from the heart and going into the gills. Now you have deoxygenated blood coming from the heart and picking up oxygen, but it's all arteries. And then you have a little capillary bed in the gill tissue itself where this is happening. Yeah, great question.
We had a question in the chat that relates back to the teeth one again.
Absolutely.
Could it be, besides habitat and time periods, why some shark teeth could be more abundant than others because of single use?
In terms of being abundant in like the fossil record, or okay, yeah.
Perhaps because like more commonly found if you're looking for teeth on a beach.
I see. Yeah.
I guess.
Yeah, commonly found there. So there's the factor of, constant tooth replacement. There's also the factor that some shark species are more abundant than others, especially, at different times throughout history. And now currently, some species are critically endangered. Many aren't. Actually, sharks are doing okay. There are some species that are not, but there are some species that are doing well. And so that dynamic of how many individuals are actually around is going to affect how often, you might encounter these teeth. Yeah, great question. Yeah. So, in terms of the idea that sharks have to keep continuing swimming to breathe is not a bad idea. That's a pretty good way to breathe, although it does take a lot of energy. It produces drag to constantly swim around and keep your mouth open, but not bad. But the reality, and we've known this for a long time, this is not necessarily new or surprising, but only a small number of sharks are obligate ram ventilators. Obligate ram ventilators means they must swim in order to ventilate their gills. So ventilation means just pushing water over your gills, whereas most sharks use some combination of ram ventilation and pump ventilation. And so this is a animation of pump ventilation, and this is based on a raven fish, but sharks is very similar, but except they've got 5 of these little gill chambers on each side of the head instead of just 1. But the idea is the same. With pump ventilation, you're able to draw water into the mouth by expanding the mouth. So you use negative pressure, and then you expand the gill chambers to fill those chambers and draw water over the gills. And then you switch over to positive pressure. So you start closing the mouth to force the rest of the water over the gills. And then you use positive pressure again to force water out the gill opening. So that's the cyclical expansion, compression, where you're switching between positive and negative pressures to maintain that movement of water over the gills. Now there's this one phase where there's flow reversal, because when the mouth is starting to expand again, there's a possibility that because water is always going to move from areas of high pressure into low pressure. So there's a little bit of possibility for reversal when they have to switch back. But fish and sharks keep that phase very, very short. So we think that it's probably inconsequential. And so this is, I love this pumping process. This is like, what I wrote my dissertation on and how this process varies across different species. It's very complex. There's a lot of different bones and muscles that are involved with actually carrying this out, as opposed to just opening your mouth and swimming through the water. And it's true that most sharks, and I would say probably all sharks, are capable of doing this pump ventilation. The question is, do they also have to have RAM in order to survive, or can they get by just on pump ventilation? And the reality here is that there's only a small number. I think there's definitely great white shark, mako, and whale shark, probably the basking shark too. There's like, maybe a couple other species, whereas there are thousands, or there's over 1,000 shark species. And so this is kind of, to be completely reliant on RAM is very rare, where we have this beautiful pump process. Mhm. In terms of the control of the pumping? Yeah, so it's controlled by a nuclei in the brainstem that like controls like all like the basic kind of heart rate, breathing rate, everything. So it's like there's a central pattern generator that kind of pushes out the signal to constantly pump. And that's a great question, because that will come up a little bit later in one of our experiments. Yeah, so it's completely, it's kind of, it is very like innate. Yeah.
Or autonomic nerves, because they're not at all like that. So.
Well, they do have an autonomic nervous system.
Not,
yeah. There's some regulation there. Like if they have like a a fight or flight response, they will increase this process. So there is feedback, there is crosstalk there, but it's not like necessarily like controlled by that system. But like the control of this is a very interesting topic. Any other questions? Pump ventilation is great. And actually, this is probably a good place to mention. That control, but just briefly, so this was a series of experiments that we did because we know, based on previous studies, that obligate RAM ventilation is very rare, and obligate RAM ventilators are pretty well studied. So we wanted to study a classic pump ventilator, and we wanted to see if we could get it to switch to RAM. Because in addition to this idea that, you have obligate RAM ventilators, and then, most sharks will do RAM as well, we're kind of skeptical of that idea because we've never seen a shark species switch from pump to RAM. And so we took the Pacific spiny dogfish and used pressure transducers. You can see them implanted in each one of the gill chambers. And this work was done by myself and Sarah Arnett, who just got her PhD from Northern Arizona University. And I love that I took that picture. I think that was an undergrad, but she was wearing the Jurassic Park, so it made me think of you guys, Fossil Club. She's a big biology nerd, just like all of us. And now she just started a postdoc at Chapman University. So this was her undergrad work. And actually, yeah, so she started as an undergrad, but she's continued these experiments throughout grad school. So we've had a lot of fun working on this together. And so with this particular experiment, we had the sharks in a flow tank, and so we could expose them to different types of flow. So rather than having the shark swim around in a track or something, we put the shark in a viewing window, and then we were able to give it certain speeds of water that it had to swim to keep up with. So kind of like a fish treadmill. And yeah, exactly like a treadmill. And so this video I'm going to show you is a kind of characteristic example of what we saw at the highest speeds. We were hoping against hope that it would switch over to RAM. But what you're going to notice is that control, that central pattern generator, that urge to pump, that's just so inherent and innate, never quit. So we saw pumping throughout the whole time we were doing all of our experiments. We never saw true RAM ventilation where they're just opening their mouth and letting the water flow. So you can see the pumping, you can see the spherical in the mouth, and the gills are kind of fluttering. And then at the end, this was like the moment where we saw this a lot, where all of a sudden it would just say, okay, the water's too fast. I have to give up now. And so then we turn the machine off and everything. But this was as fast as we could. So this was as fast as we possibly get the shark to swim. And we never saw, and we tested, I think, 6 different sharks this way, and we never once saw a switch to RAM. So that urge to pump was just pretty much constant. We never saw a situation where the shark would stop pumping and just open its mouth and let the water flow. And so we're still working on the publication here, so this is unpublished, but we are getting ready to submit it. So this is one of my favorite graphs. This is the pressure recordings of 3 different chambers. So 2, 3, and 5. So sharks have 5 gill openings. So anterior, more anterior would be 2 and 3, and then posterior would be 5. But the main thing here to note is that speed is increasing as you move from left to right on this slide. So the first panel here are the 3 gill chambers in kind of slow moving water. And then as we cranked up the speed and the shark had to swim faster, we also saw an increased amplitude in the pumping. So if we saw a switch to RAM, we would expect a complete shut off of the pumping system. But we did see the data was very messy. And we do often get things like electrical noise, but we filter out the electrical noise and we still got a very messy signal only when the shark was swimming fast. And so we think that is kind of the fluttering of the gills as water is being forced over. So there is kind of a RAM thing going on here where water is getting forced into the mouth and over the gills, but the shark is also pumping as hard as it can. You can see the amplitude of these pressures is very high. So we got a lot of positive pressure, we got a lot of negative pressure, and so the shark was really, really pumping hard. It never sort of gave up and just switched over to RAM, even though there was a lot of water kind of flowing over the gills. So that is, I consider that kind of myth busted. So it's like I said, it's not really an open question. We know that most sharks do RAM ventilation, very few are obligate. Most sharks do pump ventilation, very few are obligate RAM. And one thing that I thought was interesting while I was putting together this talk, I saw that there was a new paper that came out earlier this year where this gray reef shark, who was thought for years to be a strictly obligate RAM ventilator, they cannot stop swimming or they will drown. And it's true, we do talk about drowning in fish. But if you're not getting enough oxygen, that is one way to think about it. But then he published a paper. We just saw a paper come out where this aquarist found that their sharks would regularly engage in this behavior where they would just sit there and do nothing. Wouldn't you love it if somebody wrote a paper about you just sitting there doing nothing? Question?
So as you crank up the speed of the water, it gets more turbulent. Does the dissolved oxygen level start cranking up with it?
Could you repeat the question?
Yeah. So the question is, as we crank up the speed and the water gets more turbulent, does the oxygen level increase? That's a great question. So none of these studies we directly measured oxygen. In this system, we do have flow straighteners so that as the water is hitting the shark, it's more or less laminar. It's probably not perfectly laminar, but we have these long, skinny tubes that the water flows through right before it hits that the business part of the tank. But overall in the system, there's probably the speeding up of the motor probably does create some turbulence that could mix in more water. This water is being pumped straight from, so we have a flow through system. So this was our Friday Harbor Labs. I don't think I mentioned that. We have a flow through system where water is going directly into the tank and then directly out to the ocean. So it's probably pretty at its capacity for oxygen. So I don't think there's much in the way of fluctuation in terms of the oxygen that the sharks are getting during the experiments. But you're probably right that is happening to some extent.
Maybe converse to the question is, shark is in really polluted water with a low oxygen level, right? A dead zone. Would that influence this pumping versus RAM behavior?
Yeah. Yeah, no, that's a good question. I'm not sure. I will in the next set of experiments, I will talk about a kind of modulation where they can kind of change their behavior based on the conditions. So I might kind of answer your question about that, but we haven't, I don't think there's been any studies about water quality and breathing that I'm aware of in terms of shark ventilation.
You should do that study again.
Yeah, and certainly temperature. So temperature is going to drive drive photosynthesis, and it's going to create dead zones and stuff. So as we see rising temperature, we're seeing all sorts of issues with water being not well oxygenated, but mostly in freshwater, I think, because the ocean is so big. But certainly water quality is going to be more and more of an issue as climate change and human activity becomes more of a factor. Anything else? All right, so I think we could probably call it myth busted. So even sharks that we've seen that we know are ram ventilators, we can still be surprised and find out that they can do very well, even if they don't swim around constantly. So who knows? Someday we might do away with the idea of ram ventilation altogether. Probably not, but who knows? Or obligate ram. Certainly ram ventilation we know is happening. All right, so I want to switch over to another mystery, which is the fact that sharks have 5 gill openings on either side of the head. So they've got 5 on the left, 5 on the right. And this is kind of unusual. So we see in other groups of fishes, like the close chondrichthyan relatives, the holocephalans, which are on the kind of left side of this graph here, they only have 1 gill chamber on each side, and that's also true for ray fin fish as well. They've just got a left and a right chamber, whereas sharks have 5 chambers. And we do think that that is the ancestral condition. It's really hard to know because not only do sharks not fossilize very well because of the cartilaginous skeleton, but also we don't have the soft tissue, because you can have 5, and we do see this. This is how the condition in ray fin fish. You have 5 gill arches, but they're all kind of packed into 1 chamber. So if you want to know whether ancient shark ancestors had 5 gill chambers or 1 big chamber, you would have to have the soft tissue in addition to the bones. And so we have such few really well preserved skeletons to be able to make those kinds of inferences. But we do know that this condition of having 5 gill arches has been around for a long time. This particular paper was about these red structures, the structures that are labeled in red, which are structures that these branchial rays that support the gill tissue. And you can see in the holocephalans, those rays become a really important part of forming the gill chamber. But in sharks, we have 5 distinct chambers that we can see in the definitely in the extant species and probably in their ancient ancestors. That was almost certainly the ancestral condition for all chondrichthyans. And so the answer is why. So why would you have 5 when 1 would suffice? And that's definitely a mystery that we don't really even have a great answer to, but we've gotten some good insights from the work we've been doing. So again, this is Sarah's now into dissertation, and now she's completely finished. She's done a couple other cool shark breathing projects, a lot of like anatomy, morphology, a little bit of physiology. But this project was looking at the pressures of across the 5 gill chambers. And because of our setup and the anatomy of the shark, we decided to just stick to gill 2, gill 3, and gill 5. And so that gave us a pretty clear picture of how the pressures might differ across the different gill chambers. So we use pressure transducers. So there's Sarah implanting her pressure transducers in a shark. And again, we use specific spiny dogfish. And what you could see is that we saw a very consistent and pretty substantial drop off of pressure going from anterior to posterior. So the more anterior gills were higher pressure, and then by the time you get to gill 5, the pressures are quite a bit lower. So what do we make of this? So here are two example graphs. As much as I love the statistical graphs, I just love to see the actual pressure traces where you can really get a sense of what might be going on. And so we see in both of these diagrams on the left and the right, you can see that the second gill chamber has the highest amplitude of pressure. So the overall up and down of the positive and negative pressures. And then gill 3 had the kind of a intermediate, and gill 5 had very low pressures. And on the left, figure A here is what we consider kind of normal breathing, like the standard breathing patterns that we saw in most of the sharks. And then on the right, part B is what we considered quiet breathing, where we could notice the shark slowing down its breathing. And so this is where I was talking about, potentially modulating. So there's some behavioral stuff going on here, potentially some stuff with the autonomic nervous system. So, rest and digest versus fight or flight. So when the shark is able to kind of settle down and breathe in a very quiet pattern, maybe they have plenty of oxygen, the water quality is great, they don't really need to do their normal breathing pattern, they can kind of not just slow things down. And you can see the rates here. Actually, the one on the right, the quiet breathing is actually a little bit faster if you look at the time scales. These are 10 breaths each. Was able to kind of lessen the amplitude of the pressure, so it didn't have to do as much pumping. And then gill 5 was effectively inactive. You can see a little bit of a pattern there, but these pressures are low to begin with. 200 pascals, not high pressure. There's very, very low pressures. We usually measure, like for human applications in engineering, we're usually measuring in megapascals, and so or kilopascals at least. So these are very, very small pressures to begin with. And this one on the right here was kind of the gill 5 was actually pushing like the capability of our pressure transducers to even detect anything. So probably the pattern here is probably maybe little bits of water getting into the gill chamber. We don't think that this is, like from the rest of the pumping cycle, we don't think that this is active breathing. And what we were able to see from externally from our video is that basically you don't see pretty much anything happening with gill 5 during quiet breathing. So that's really interesting. This is the first time we're documenting kind of a behavioral modulation where the fish can, depending on its condition, have maybe save some energy by having a lower amplitude and not using all 5 gill openings, which was a really exciting finding, very interesting, and it gives us a sense of why you might want to have 5. So you have 5 if you need them, but then you have the option to be able to turn some of them off when you don't actively need to maximize your ventilatory capabilities.
Data normalized water.
Yeah, so we also had, in each of our trials, we had an ambient, which I think you could see the ambient in. So one of these, so there's 3 here, but then the 4th one right there externally is an ambient pressure. So we were measuring ambient pressure, which was especially important for these guys swimming. We were always correcting for what the ambient pressure would be on the outside. Any other questions?
So the way you describe the blood flow.
Uh-huh.
Is gill 5 just shutting down or low activity because it's not exposed to highly oxygenated blood?
I think it's probably you've hit on something really interesting, but it's kind of the other way around in the sense that they will we think that they perfuse less of their gills when they're quiet breathing, and there have been some studies where they're measuring directly oxygen and blood perfusion in ram ventilators, and they found that sometimes the sharks aren't sending blood to all of their gills. And so it's more about it's like a chicken and egg thing. I think it's more that when they're doing their quiet breathing, they may also be, we don't know for sure in this case, they may also be kind of shunting blood away from the gills, so that they're kind of perfusing less of the tissue where they're not going to be pumping. That's just a hypothesis for this particular situation, but we have seen like different levels of perfusion, or other researchers have seen different levels of perfusion depending on the conditions in different species of sharks. Yeah.
What's the difference in volume of water moved with these two different activity levels?
That's a great question. So to get volume, we need both pressure and velocity, which we don't have. So we can't calculate it necessarily very easily. But yeah, I don't think there's been an attempt to do volumetric flow rate in shark breathing. There's been kind of like simulations done in fish breathing, but even that's very difficult because there's a lot of factors that you need to consider when looking at volume change that are hard to do in a kind of a live animal kind of scenario. So there's been like modelings and simulations, but never directly measured. Yeah, it doesn't give you water speed. So, and you can use like particle tracking. Like if you seed the water with some neutrally buoyant particles, you can like look at the flow rate out of the gill openings, but that doesn't even necessarily tell you what the flow rate is internally, but it can give you like kind of a sense. Like there are ways to get at it, but it's kind of a really hard thing. Like I would love to have, flow rate and pressure at the same time. But flow rate is a little bit more difficult instrumentally to get something that works in the water. But yeah, in an ideal situation, we'd have all that data right at our fingertips. Yeah. Yeah. Any other questions? Great question. Thank you, everybody. All right. So we've kind of got an answer here. So if you have more gill openings, you have the potential to modulate in ways that ray-finned fish can't. And also another thing, so this is a robotic shark, so I'll play this video. We haven't tested this yet. Michael just finished building this in the spring, and then we haven't gotten around. So we need a version 2.0. It's not great, but we want to do a robotic model of gill ventilation in order to answer the question: is the decrease in pressure, specifically the drop off in pressure that we see in the normal breathing, is it a passive or active pattern based on fluid dynamics? So there's a possibility if we do a model where we would see so we wouldn't model the gill musculature. So we need to like mount little chambers on here, but the chambers wouldn't be actuated. And so we would just measure pressure inside of them. And we might expect that we would see a drop off in pressure just by the fact that pressure might attenuate when the mouth is doing its pushing and the water's moving further and further back in the throat. There may be a drop off in pressure. So this is just a cool, shout out to Michael who graduated. So I'm going to need another student to work on this. He did a really great job getting this thing to at least kind of look like a robo shark and move like a shark. But we're not quite there yet. But the idea with this is that we want to test this question: is this pattern that we're seeing of a drop off of pressure, is this because the sharks are actively using the gill chambers less and less, or is there also a passive fluid dynamic thing causing that drop off? And I predict that it's probably both, but we won't know for sure if there's passive effects until we do the modeling. But we know for sure that they can turn off gill 5. But whether the fact that we always see a pattern of pressure drop off is passive or active is kind of an open question. So we also do some robotics in the lab. We have one of my other projects in addition to breathing is studying fish burials. So we have a burial robot as well, which works really well. This is kind of in its beginning stages, but it's also really cool. It's always nice to make a model. If you've got questions that you can't answer, then build a model is always a good strategy. I just love that little guy. All right. I love the eyes. Once you put eyes on something, then it feels real. All right. Yeah. So that was just a little senior project. Okay. So that's the shark breathing. But one of the things that we've also been thinking about over the years is, can we flip this question on the head, and instead of asking why do sharks have 5 gill openings, ask why did chimera evolve 1? Because 2, a left and a right, but one on each side. And because we think that the ancestral condition was 5, and chimera, which are close relatives of sharks, and also ray fin fishes went to a 1 gill kind of scenario. And so this was a project done by Jarila Kamau Wang, who recently just got her master's degree in marine science from Northeastern University, and she's now working for Ocean X, which is a really cool industry partner for ocean science. And there she is doing her pressure transducer surgeries on these little chimeras. Very cute, depending on who you ask. But they were great to work with. And the cool thing that we found about this study, and I'm not completely sure, this is also unpublished. We never published this for various reasons. But it's a really cool data set, not only because we saw really interesting things with their breathing, but what you're seeing here on this chart is actually breathing with coughing mixed in. So the big peaks are coughs. So fish can cough, and that's like another. If I had to say, like, one of the biggest mysteries is there's a huge mystery as to why and how fish are able to cough. And they do it in all different types of scenarios. One of the most common hypotheses is probably correct in a lot of cases, is that they're clearing gunk out of their gills. And so this is a flow reversal. So that whole pumping process that I talked about earlier, it's just basically that, but completely reversed. So we see the pressures flip the other way around. And we also see really high pressures in the gill chamber to force water over the gills back into the mouth and out the mouth. And we were able to see, I like this video of the fish on the sand because we had some sand in there to keep the fish happy. And we also were able to see every time it coughed, we were able to see a nice big puff of sand get disturbed. And so we knew for sure that we were seeing a coughing behavior. And these pressures are extremely high for what these fish are normally capable of. So they're usually pretty quiet breathers, but then we were seeing pressures going up to over 1,000 pascals. And so this is much higher, not just than the normal breathing of a holocephalin, but also this is the most dramatic cough I've ever seen from a fish. Usually, like, if they get above 500, it's impressive. Well, depending on the size of the fish, of course. But for a fish this size, this is a really, really impressive cough. And they were doing it repeatedly. And it didn't matter which individual we looked at, this was a pretty common behavior. At some point or another, we would see them start to do this kind of coughing behavior. So my general, this is just completely, I was thinking about this over the past couple weeks and talking to Jarila and Sarah if we can collaborate, because I think Sarah has some coughing from her sharks as well, which we threw out for our first papers, but now we might go back to it. The coughing for the sharks was much less dramatic. And these holocephalins are duraphagous, which means that they crush their prey. So they're eating things like clams and mussels, little bivalves, and so they're spending a lot of time chewing and crushing in their mouth, and potentially that's going to get a lot of stuff gunked up in their gills. Whereas, especially the dogfish, they're pretty much just swallowing things whole. They're not doing a lot of processing. And so we think that potentially, this is just like completely off the dome, but that having one big gill chamber allows them to clear their gills much more easily if they can just do a nice big cough, get everything cleared out every once in a while, and make sure that their gills are free from various materials. So that's just one potential hypothesis as to why chimeras and ray fin fish have gone towards this one gill opening, especially because in ray fin fish, they've got a lot of bony structures that support the gills, whereas in the chimera, it's pretty much just all muscle. So they've got just one big bag of water that they can just squeeze and just force out the gills and clear anything that might be stuck in their gills. One of the reasons we think they were doing it so often for our study is that they probably were trying to get rid of the pressure fins. So yeah, because I mean, we've never seen really any distress or anything in these animals when we're doing our studies. But if these guys are sensitive to having things in their gill chamber, this might be a normal reaction for them to say, all right, I need to work on clearing this stuff out. All right, so that's just another project that kind of ties in with our mystery solving approach to these bigger questions about shark breathing. Any questions about that? All right. So just to kind of wrap this up, sharks, we don't know for sure why they have 5 gill openings, but it's possible that this helps them. You know, we've seen some pretty clear evidence that they are able to modulate their breathing. And, but there's also this, you know, very common idea in evolution where if you have something that works, why get rid of it? So we know that 5 gill openings is probably the ancestral condition. And if it was working out great for their ancestors, why not just keep it around and maybe play around with it a little bit? But if it's not broke, don't fix it. So if there's no reason for them to switch over to a different type of breathing or a different type of morphology, then evolution's not going to favor that. And then also, like I just mentioned, chimera may have only 1 gill opening or 1 gill chamber to produce these large coughs to clear their gills. That's just a hypothesis. And of course, with all of these studies that I've showed you, the reality is always it's complicated. We don't know. We always end up at the end of these studies with more questions than answers. And we also, you know, it would be great to write papers where we have everything figured out. Like, I would love to, it would be so much easier to write if it was just like, yeah, we just solved this. It was very, very clear pattern, very easy to understand what's going on. But whenever we start doing any one of these studies, even if we think it's a no-brainer, easy study, we're gonna, you know, our hypotheses are very clear. We always end up with a really interesting story at the end that tells us that things are probably more complicated than we thought when we set out to do the work. And I also just want you to take away something else about how we think about evolution and the complexity. So when we think about evolution, the word adaptation often comes to mind, where, you know, and we've talked a lot about adaptation today, where we think there's some sort of beneficial change to the morphology that can make the system better, and evolution is going to favor that. And so over time, you would see an adaptation, new morphology, morphology used in different ways. But also there are a lot of other factors that go into the evolution of structure and function. One of my favorite is multifunctionality. So a lot of structures perform more than one function. So not only is the breathing system for breathing, but also because it involves the mouth so heavily, the mouth is under selective pressure for things like feeding. And even in sharks, you know, they often will even use their teeth to hang on to the female while they're mating. So there's even a role for the jaws in the mouth during mating. So there's lots of different functions that the head has to perform other than just ventilation. And that's true for most morphology. There's usually more than one function for a structure. Exaptation is when a structure takes on an additional function that it didn't have before. So think about bird wings. So dinosaurs had these tiny little forelimbs, and then eventually they evolved to have feathers on those forelimbs, and eventually they could use those little forelimbs to actually fly. So they were able to evolve wings from their forelimbs. So that's exaptation, using something that's already there. There's also historical constraints. The idea of if it ain't broke, don't fix it is something you're going to inherit things from your ancestor, and that's going to constrain what you can look like in the present. And then, of course, there's random events like the asteroid wiping out the dinosaurs. Maybe we'd be a society of dinosaurs if that hadn't happened. You know, there's randomness too, all around us, and it's slow environmental changes too, like climate change, not just now but in the past. Things change; they don't stay static, and so all of these different factors affect evolution. And the factors on the right are things that affect our ability to understand evolution. So, of course, you're all probably very familiar with the fact that the fossil record is incomplete and imperfect. And so you can't just go to the fossil record and get all your questions answered. And also, we can't just go ahead and repeat the experiment of evolution. So evolution happened, you know, or it's happening, and it happened in the past. But we can't like glance into the past and see things as they were happening in the past. We can only make inferences based on what we see today, whether it's fossils or whether it's looking at the living creatures. And then also, if you have extinct animals, you can't study their physiology and behavior. So you have to make inferences there as well. And there's also the fact that science costs time and money. I don't know if it might have occurred to you that the questions that we asked today are questions that have been around for a real long time. Why has no one answered them? Probably because, you know, it takes a lot of time and money to do these kinds of things. And hopefully Lisa Whitenack has her dynamic testing system, and then we have all of our teeth questions answered. But it takes a lot of time and money to develop those tools and to actually carry out the work. So it's always complicated is my main take home. And I want to just briefly get serious. I always do this at the end of my talks, and especially now at this time, it's very important. You're all here because you're interested in science. And I want you to think about the complexity of the stories that we talked about today and how evolution is complex in general. And just keep an eye out for people telling you overly simplistic stories about, especially about human behavior. So this is very common. It used to be less common. Now it's kind of ramping up again, where people are using evolution to justify having racist, sexist, and homophobic and other types of beliefs that are, they try to prove it with science, but it never holds up to scrutiny. So this is just a brief trigger warning for, on the next slide, just very briefly, I'm going to show you some paper titles that cover these topics. So if you read this, these are all papers where the science itself has been analyzed and debunked. These are people with an agenda, people who want to tell you that humans are tribal, that it's inherent that humans are going to fight each other and have these big wars, that that's a part of who we are. That is not true. Also, the idea that different races of people, if you read this closely, which I don't recommend, that we have, we know race is a social construct, and we cannot tie differences in behavior to race, first of all, because it's not biological. Second of all, because even the work presented in here, this terrible person here, Philippe Rushton, actually used Penthouse magazine to do his quote unquote research. Yeah, it's kind of funny until you think about it a little bit more. And then this paper was about how when women are ovulating, they change their clothing, and that had a lot of methodological issues. The scientists just had an agenda there. Again, this last paper here was about why the women who are homosexual exist because they don't want to compete with other women, and we know that that is not true as well. So what I want you to do is just, as you're going around in your daily life, be skeptical. You're all here because you love science, love evolution, and you love thinking about these creatures and our history, our shared history. And so you have all the tools that you need to be skeptical. When you see something where there's an agenda, the science doesn't hold up, you can look at the science, you can look at their methods, and I just encourage you to always be skeptical. If you see a headline, maybe look into it a little bit more, but just, I want everybody in my life and in the world to just be skeptical about evolutionary stories, because at the end of the day, it's always complicated. And these simple stories are almost always not true. And with that, thank you so much for having me out tonight. I had a great time. This work is funded by the National Science Foundation, Friday Harbor Laboratories, and my home institution, Howard University. And it couldn't have happened without my collaborators, Adam Summers and Laura Ferry Graham. And I really appreciate the Natural History Society of Maryland having us out tonight, and for you guys for showing up. So thank you very much.
Thank you.
Question? I know we've been taking questions throughout, which was great. Yeah, thank you so much. Yeah. So I probably should have. So the marine labs where I do this kind of work is really important to acknowledge, and I don't think I did a good enough job. So Friday Harbor Labs is a marine lab in Washington State that is on an island about 2 hours north of Seattle, and it's a beautiful place, wonderful place to take students. I used to take students there every summer. Now I'm going to be starting officially next summer, taking students to Woods Hole, Massachusetts, because it's a little closer. They have a little bit more of the equipment and stuff that we need, but I love Friday Harbor because it's just got this like fish summer camp vibe. It's a great place. The people there are wonderful. I mean, just having Jeff Bass as the head engineer, just be able to just whip us up a quick device was incredible. And so like doing this work at Marine Labs is great because also they've got flow through seawater, so you're able to keep the animals happy, in good condition, right next to the water where you can pull them out of, so you don't have to transport fish everywhere. And students just love to come and be immersed in the marine environment. Yeah, so this is Washington State, and it's operated by the University of Washington. Yeah. Any other questions?
Thank you very much, Stacy.
Yeah, thank you. Thanks so much for having me.
How many students are in your program?